Okay, let's go ahead and start with chapter 9 and genetics. And I have broken this chapter up into three sections. The first section, part 1, is going to be DNA and DNA replication. Part 2 is going to be protein synthesis and the mechanisms of transcription and translation. And then part 3 is going to be gene exchange and mutation and how bacteria and other microorganisms gain diversity in their populations. So let's go ahead and start talking about DNA replication. The first thing that we need to do is we need to understand what genetics is. Genetics is the study of inheritance or heredity of living things. How do we transmit traits from one parent to the next generation to their offspring? Genetics is also the expression and variation of traits. We're going to find that all organisms are unique in their traits, even if they are organisms of the same species. They are still going to have some variation in the expression of their traits. We're going to look at the structure and the function of genetic material and what is the genetic material of living organisms, and how is this material changed? How do we gain change in our genetics? Because if we don't have diversity, remember that once you eliminate diversity or variation in the change of genetic material, you lead to the elimination of that particular organism. We can have we can look at genetics on several different levels. We can look at it organismally, we can look at it chromosomally, and we can also go clear down to the molecular level and look at it as a molecule. The nature of genetic material is very specific. The first thing is it has to be able to be self-replicated. When we start to go through cell division, this DNA has to be able to be replicated and be replicated accurately and completely so that it can be separated from the parent into the daughter cells, and DNA replication and cell division then can be completed, and each daughter cell now is completely identical genetically to the parent. When we look at the levels of structure and function of our genetic material, we have three different levels. We have the genome, we have the chromosome, and we have the gene. When we talk about the genome, this is the sum total of the genetic material of the cell. This is every single solitary nucleotide, every gene, every chromosome that we have. Our genome is structured mostly in our chromosomes. We have 46 chromosomes for us, for humans. Other organisms have more, some have less, but most of the genetic material is going to be housed in chromosomes. You can have some non-chromosomal sites of genetic material. You've got plasmids, you've got the mitochondrial DNA, you've got DNA in the chloroplasts of plants. So not all of the genomic DNA is going to be in chromosomes. You do have some of it that will be extra chromosomal. In living systems, in cells, the genetic material is exclusively DNA. We have RNA as an intermediate molecule, and you'll see when we start talking about protein synthesis and transcription and translation that RNA is a very important molecule, but it's an intermediate molecule from the storage of DNA, or the storage of information in DNA, and the expression of that information in proteins. So when we're talking about our genome, the material is exclusively DNA. Here's a great picture that's in your book, figure 9.2. It shows you all of the places where we can have our DNA. We can have it here in the chromosome, whether it's a eukaryotic chromosome that is a paired linear multiple chromosomal molecule, or you can be over here in the bacteria or the prokaryotic cell where you have a single circular chromosome. We do have plasmids in things like fungi and protozoa, as well as the plasmids that we see here in bacteria. Then you also have some of this extra chromosomal DNA in the mitochondria, as well as the chloroplast that you see in plants. So you can see all of the places where we would normally see genomic DNA. From the genome, we go down to the chromosome. The chromosome is a discrete cellular structure that is composed of DNA that is very neatly packaged so that it can fit inside the nucleus. You're going to see that DNA is an extremely large molecule, and we need to be able to package that into a small, tiny space like the nucleus. In eukaryotic chromosomes, the DNA is going to be tightly wound around some proteins called histones. And as we wrap this DNA around histones, it's going to condense it smaller and smaller and smaller so that we can now fit it inside the nucleus. Most eukaryotic cells can have chromosomes that are paired. This means that they are diploid, or you can have single chromosomes that are haploid. These eukaryotic chromosomes appear linear, and there are differing numbers of chromosomes, from very few to hundreds, even thousands of chromosomes. If you look here, you'll see that the fruit fly, Drosophila melanogaster, has 8 chromosomes. Everything that fruit fly needs is housed there on those 8 chromosomes. Humans have 46 chromosomes. Some species of orchids have 108. And you have a particular plant called an adder's tongue fern that has 1,260 chromosomes. So very complex organism. Bacterial chromosomes are also condensed and secured, but they are not wrapped around histones per se, but they are wrapped around histone-like proteins. They have much the same structure, much the same function, but their amino acid sequence is completely different. So they are not histone proteins, but they are like histone proteins. Instead of having multiple linear chromosomes, bacteria have a single circular chromosome. Once we leave the chromosome, we're going to go down in structure to the gene. All of our chromosomes are made up of genes. All of our genes are held on our DNA, and a gene is a certain segment of DNA that contains material that will be used to code a protein or some sort of RNA molecule. We can divide our genes into structural genes or regulatory genes. Structural genes are going to code for specific proteins that are going to give us a specific expression. Regulatory genes are going to be genes that control gene expression or protein synthesis. You're going to find that when we get into protein synthesis and gene expression are the same thing. The sum of all of our genes in an organism is called the genotype, and the expression of that genotype is the creation or the expression of traits, and that is our phenotype. All organisms contain more genes in their genotype than their phenotype requires at any given time. So a good amount of the DNA on any organism is going to be junk DNA. If you read through our book, and actually if you're reading through your A&P book, one of the things that it talks about is the amount of codable information versus the amount of uncodable information. And the human genome has more junk than it has codable information. You're going to see a great variety in the size and the packaging of genomes. You can have genomes that are as small as 4 or 5 genes and things that are like viruses, to human cells where you have 20,000 to 25,000 different genes in a particular genome. If we were to take the DNA out of any cell, whether it's prokaryotic or eukaryotic, that DNA can be stretched out and will be about 1,000 times larger than the cell it came out of. So we're sitting here packaging up tons and tons and tons of molecular matter and having to put it in a very, very small space. This next slide is going to show you a picture of an E. coli cell. This yellow cell here in the middle is an E. coli cell that has released all of its DNA out into the extracellular space. This is one great big circle. If we were to be able to pick this up and lay it out on the lab bench, we would actually be able to see it. It would look like a very fine hair. But all of this DNA is going to have to be wound up and condensed and put inside this small little space. So you can see that we can take this molecule and condense it down very, very tightly and secure it inside the cell. the code for DNA is actually a very simple code. It's a matter of 4 nitrogen bases that put in specific sequences can make the information available for the coding of proteins. In 1953, 2 gentlemen by the name of James Watson and Francis Crick were the first ones to discover the structure and the type of molecule that DNA is. They discovered that it, number one, it was a nucleic acid, and number two, that it was made up of 2 strands, and that those strands form a double helix. You've got one helix winding around another helix. They were able to find this information based on a picture that they saw from another scientist by the name of Rosalind Franklin. And she had taken an X-ray of DNA, and they could tell by the shape of the X-ray that this was a 2-stranded molecule that crisscrossed along the way because of the helices that would one right on top of the other, and it showed kind of an X in the center. Unfortunately, Rosalind Franklin died before she could get any of the credit for her part in the discovery, and so unfortunately we know that Watson and Crick are the 2 main players in the discovery of DNA. The base unit of DNA is the nucleotide, and when we have a nucleotide, you're going to have your phosphate ion, you're going to have your deoxyribose sugar, and you're going to have your nitrogen base. When we look at this deoxyribose sugar, we need to be able to count the carbons, because it's going to be important that we understand where we're sitting as far as this sugar is concerned. When you look at deoxyribose, and unfortunately they didn't include the oxygen here at the top of the sugar molecule, we're going to move to this first carbon here where the nitrogen base is attached, and this is the number 1 carbon. When we come down to the second space, this is the number 2 carbon, this would be the number 3 carbon, this would be the number 4 carbon, and attached to the phosphate is the number 5 carbon. When we start looking at how the nucleotides bond to each other, those number 3 carbons and those number 5 carbons are going to become very, very important. When we take our nucleotides, you'll see that we have 2 nucleotides here. We have nucleotide 1 and we have nucleotide 2. And to bond these together, we need to be able to dehydrate them. I'm not going to have you know how to dehydrate and synthesize the bond between the 2 nucleotides, but you do need to understand that by bonding the number 3 carbon of the first sugar, of the first nucleotide, with the phosphate of the second nucleotide, let me get my cursor back here, there he is, with the second nucleotide, we form a phosphodiester bond between the sugar of the first and the phosphate of the second. This is a very strong bond that gives us this phosphate sugar backbone that will protect the nucleotide center that we have, or the nitrogenous base center that we have. And we're going to find that it's the sequence of these bases that becomes very important. And it's the sequence of these bases that gives us our amino acid sequence to give us our protein structure. And what do we know about structure? Is that structure determines function. Our nitrogen bases in the nucleotide can be divided into 2 different categories, and we talked a little bit about this in unit number 1, but I know we've all slept since then, so let's go and review it a little bit. We can divide our nitrogen bases into purines and pyrimidines. We have 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. The purines are adenine and guanine. The pyrimidines are cytosine and thymine. When you have your nitrogen bases, they are going to bond to each other as a complementary pair. Each of these nitrogen bases on a single nucleotide is going to be bonded to the number 1 carbon of the sugar. Remember, we counted those carbons, and when you count them, at that number 1 carbon, that's where your nitrogen base will be attached on that nucleotide. We talked about the shape of DNA and that it is a double-stranded molecule. The pairs of nucleotides will always be 1 purine bound to 1 pyrimidine. Always, always, always. This way we get good structure, of our DNA molecule. It's very strong, it's very rigid. The complementary bases aren't just purine to pyrimidine, but a specific purine to a specific pyrimidine. If we look at it, we will see that adenine always, of one strand, always binds with a thymine on the second strand, and guanine always binds with cytosine from one strand to the other. The paired bases are going to be joined by hydrogen bonds, and we join these with hydrogen bonds because they are going to be easily broken. Because when we go through DNA replication, one of the first things that we're going to have to do is we are going to have to separate or unzip the molecule so we can get to those nitrogenous bases on the inside. When those are open, then we can start replication. If we were to bond these with some other bond like a covalent bond, we would have to apply a lot of energy to break this molecule open so we could start replicating. And if we did this, then DNA replication would be an extremely energy expensive procedure. This way, by bonding the bases together at their nitrogenous bases, or at the strands together at their nitrogenous bases, those hydrogen bonds are easily broken, and we can get to the inside and begin replication. Adenine bonds to thymine with 2 hydrogen bonds, and guanine bonds to cytosine with 3 hydrogen bonds. One of the characteristics, too, of the DNA is that it has an anti-parallel arrangement, meaning that one strand runs from the 3 to 5 prime and the other strand runs the one helix turns in the 5 to 3 prime direction, the other turns in the 3 to 5 prime direction. So when you look at it, if we unwind it completely, the 5 prime end of one strand will be bound to the 3 prime end of the other strand, so that they are anti-parallel, and you're going to see that they run in opposite directions. The significance of the structure of DNA is going to be in the arrangement of the nitrogen bases. This is going to allow for the maintenance of the code during replication, meaning that I am going to conserve the code sequence. Base pairing guarantees that the sequence will be retained from one cell division to the next cell division. When these strands of DNA are separated, each strand then is going to act as a template for the new daughter strand that's going to be replicated. This is going to ensure that we have exact copying of the DNA and that my genetic material remains stable, and I have good fidelity between parent to offspring. The template strand is automatically going to determine the sequence of the newly replicated strand. If I have TAC on my parent strand, I automatically know that the new replicated strand is going to be ATC on the other, or ATG on the other strand. So one strand determines what the other strand sequence will be. It also the arrangement also provides variety. The order of bases is going to constitute basically a genetic program. The sequence of bases are going to make genes, and the collection of those genes is going to make the genome. The genes, each of us have specific genes, and we've got a lot of genes alike, but the sequence of those genes is what allows us to have some unique qualities. When we have genes in a particular order or combination, this is going to give us some unique qualities. Brown hair, blue eyes, rather than blonde hair, brown eyes. So those unique combinations are going to give us unique qualities. In other words, your unique sequence of bases is what makes you unique. DNA replication is essentially preserving the code and being able to pass it from one generation to the next. The process of duplication is going to allow us to make sure that traits from one generation are going to be given off to the offspring. Therefore, DNA replication has to be completed in a single generation. If I begin cell division and DNA replication is not completed by the time cell division is over, then I have daughter cells that don't have sequences that may be important, and I haven't maintained the fidelity or the the trueness of my genetic material. So DNA replication has to take place in one complete cell division cycle. Once it's done, the cell separates, and now I have two daughter cells that are genetically identical. Overall, the replication process is an enormous process. It requires about 30 different enzymes. We have to have enzymes to separate the strands. We have to have enzymes to copy the template. And then we have to have enzymes that are going to produce the new daughter molecules. So we have an enormous number of enzymes that are going to be working all the time, and it's quite the juggling act. I am only going to hold you responsible for about 6 of these enzymes. And one of the enzymes is a little bit different in your book than what I have here in the notes, and the ones in the notes are the ones that I want you to know. So be sure that you follow those. If we look here on this table, your book calls DNA gyrase topoisomerase 1. They're the same enzyme. I just think DNA gyrase helps you understand what it does better than topoisomerase. So please use DNA gyrase as the enzyme that you're going to remember. All of the rest of them are going to be exactly the same. The first enzyme that I want you to know is helicase. Helicase's job is to take the double strand and unwind it and unzip it. So we're going to take this helix, we're going to unwind that helix, and we're going to separate the two strands. You're also going to have an enzyme called RNA primase. One of our strands is going to require an RNA primer, and the job of RNA primase is to synthesize that RNA primer. DNA polymerase 3, this is going to be the enzyme that will add bases to the newly synthesized daughter chain using the parent chain as a template. DNA polymerase 1 is going to come in, and remember I said one of the strands is going to have RNA primers. DNA polymerase 1 is going to come in, it's going to remove all of those primers, it's going to close any gaps there are, and it's going to repair any mistakes that might still be in the DNA molecule. Finally, we've got DNA ligase. DNA ligase is going to come along this strand that needed those RNA primers, and it's going to bind any type of nick or separation that you might have in that DNA synthesis process so that you have one nice long continuous strand of newly synthesized DNA. Once you have that new daughter strand made, you've got to recoil that molecule up and condense it down and start wrapping it around those histone proteins. And the job of that is DNA gyrase. DNA gyrase will supercoil that DNA so it gets smaller and smaller and smaller, and finally will fit into the shape of the chromosome so that it can be put into the nucleus and complete DNA replication and then complete cell division. Let's talk about the details of replication. The first thing that has to happen is you've got the two strands that are going to be unwound and separated by helicase. Helicase is going to bind to a spot on the DNA molecule that is called the origin of replication. Once it finds that origin of replication, it's going to unwind and unzip these parent strands. These parent strands are also known as the template strand. Once we've unwound and separated these, now we're going to have free unbound nucleotides that are going to come up and match the newly separated nitrogen bases of the parent strand. So think about if you've opened it up, if you want to take a zipper and open up that zipper part way, now you have part of that zipper where the teeth are not attached to anything. So the teeth that are not attached now are ready to have new DNA molecules come up and start building that new daughter strand based on the sequence of the parent strand. DNA polymerase is the enzyme that is responsible for adding these new nucleotides to make this new DNA chain to fit along this template or parent strand that we're using to create the new strand. We said that the bases would always pair purine to pyrimidine. Wherever on the parent you have an adenine, thymine will bind on the new strand, and vice versa. If you have thymine on the parent, adenine will be brought in on the new strand. Wherever you have guanine on the parent, you will have cytosine bind, and vice versa. If you have cytosine on the parent, guanine will be the nucleotide that will be put in place on the daughter strand. If you have some bases that are improperly inserted, they will be removed and the correct base put in by DNA polymerase III. As it's moving down the strand, if a base is improperly put in, DNA polymerase will proofread, it will snip out the old one or snip out the improper one, put the new one in, and everything will then continue to go on. This helps to maintain the fidelity of this replication so that things stay the same from generation to generation. I do see an error here. This DNA polymerase right here, this should actually read DNA polymerase 3, not DNA polymerase 2. I apologize for that. So in your notes, be sure that you fix that to DNA polymerase 3. You have some specifics of DNA replication when we're talking about separating the two parts of the parent. When those strands separate, when they unwind and separate, they create a replication fork. If you look here, you'll see that we have the parent here that is not yet unwound and separated, and here we have one parent and one daughter. Here we have a parent and a daughter, and everything that's going on here is this called the replication fork. This is where all the nitty-gritty of DNA replication takes place. If you've gone in front of the replication fork, you'll see that DNA replication is complete. Behind it, it hasn't opened up yet. Everything, all the nucleotides and stuff are going to be being added here at the replication fork. I want you to take notice here, and I probably should have put this picture on in another slide, but I want you to take notice that we have one strand here that is continuous and is being replicated as the replication fork moves down the line in one long continuous strand. But let's look at this strand here. You'll see that it's replicated in sections or in pieces. Keep that in mind as we move down to the next couple of slides. You're going to have nucleotides added at the replication fork, and that replication fork is going to continue to move down the parent strand. It's going to open up that parent strand farther and farther as it goes down. DNA polymerase III is going to add nucleotides as they become available. You're going to add nucleotides, and you're going to then continue to make that new daughter strand. DNA polymerase can only add nucleotides to a free 3' hydroxyl on the nucleotide sugar. Remember, we talked about the 1, 2, 3, 4, and 5 carbons. The number 3 carbon is the 3' hydroxyl. DNA polymerase has to see that hydroxyl on the 3' end is open. If it doesn't see it, it won't attach and it won't add nucleotides and make the new chain. Because of the anti-parallel nature, only one of those strands will have an open 3' end. Remember, the other side will have an open 5' end. DNA polymerase will only work as it can see an open 3' end. So because of that anti-parallel nature, the two strands will move in opposite directions. Let's go back to this slide in the picture that's on the previous slide. If we look here, you'll see that one strand is moving in this direction and being replicated as the replication fork moves on. The other strand is being replicated in the opposite direction. And so it's kind of moving, instead of following the replication fork, it starts at the replication fork and moves back. The one strand that is following the replication fork is called the leading strand, and the other strand is called the lagging strand. The leading strand has an open 3' hydroxyls. Those bases, our DNA polymerase III will bind to that. Bases can be added directly to that leading strand. That leading strand is going to be synthesized continuously. It's going to follow the replication fork down, and DNA polymerase is going to add bases as it goes. The lagging strand does not have an open 3' hydroxyl. It has that open 5' end. And to add bases, we have to create a 3' hydroxyl, and that's where our RNA primase comes in. On the lagging strand, RNA primase will bind and make a short RNA primer that will give or produce an open 3' hydroxyl. When DNA polymerase recognizes that open 3' hydroxyl of the RNA primer, it will bind to that primer and begin to synthesize. And it synthesizes from one primer to the next. The lagging strand is going to be synthesized discontinuously in pieces as DNA polymerase moves away from the replication fork. So as that replication fork opens, an RNA primer is going to be made. DNA polymerase will bind to that primer and replicate to the previous primer. And so now you have small sections of DNA rather than this long continuous strand of DNA. Because it can't synthesize DNA continuously, DNA polymerase can only add a few bases at a time. DNA polymerase binds to the primer on the lagging strand and it begins to synthesize from primer to primer. Once that section has been completed, now DNA polymerase 1 will come and bind to that primer, clip out the primer, and add the correct DNA sequence in its place. Once it has taken all of those RNA primers out, now you have sections of DNA on the lagging strand, and these sections are called Okazaki fragments. These fragments are going to have to be closed up because we can't have pieces of DNA that have big gaps in them. So these Okazaki fragments are going to be attached together by the enzyme DNA ligase. DNA ligase will fuse all of those pieces of DNA by taking out the primer and forming a phosphodiester bond between the pieces and giving you a long continuous sugar phosphate backbone, and now we have a nice continuous piece of DNA, and when it's complete you will have two strands of DNA that are genetically alike. During DNA replication, you will have some misspellings, if you will, where improper bases have been put in and are not corrected. The wrong base can be added, but these mistakes are extremely infrequent. You have one about every 100 million to 1 billion bases. And so if you think about that, that's not a lot of mistakes. Most of the mistakes are going to be corrected by DNA polymerase 3 or DNA polymerase 1. Both of them can cut out incorrect bases and put the correct base in place. So we actually have two proofreading systems, so it's like two sets of eyes instead of one. If those bases are not corrected, if we end up having a sequence that is, if you will, out of sequence, then we have a mutation. And we'll talk about mutation in part 3 of this lecture. Replication is going to be completed when the replication fork meets the end of the parent strand. Okay, once the parent strand is completely replicated, you're going to have two molecules that are genetically identical. They are going to be made up of one original parent and then one newly synthesized daughter strand. Once those are completely done, the strands are going to rewind with DNA gyrase. DNA gyrase is going to wind them up, condense them, and we now have our nice piece of genetic material. Each new daughter strand is going to contain one parent and one daughter, each new strand of DNA is going to contain one parent and one daughter. And because we have kept part of our old molecule and it's been preserved, DNA replication is considered semi-conservative, meaning that it's half old, half new. Each daughter molecule is going to be identical to the parent in its composition, and one strand is going to be completely new and the other strand is going to be old. I have a set of animations that I think I have figured out how I can get them to you, but they'll come to you through an email. That's the only way I know to put them up, and instead of on the assignment page. So I will send them as an email, and I would encourage you to watch these so that you can get the details of replication down, and they're very easy to watch. I do want you to take some time to start writing down questions. Take time to go through these and fill out the study guides as you go, so that you are kind of keeping up with this whole replication process. I'm not sure what this is, but we're going to cancel it and we're just going to go on. So again, I will put those animations out there. You will be able to read them, you will be able to watch them, and this will give you an opportunity to write down questions that you have. I will take a little bit of time before we start on labs to go over any of those questions that you have. If I need to draw anything out on the board, I can do that. I know this is kind of hard to kind of put in your brain as you read through it, but I think you'll do fine. Just be sure that you kind of keep up with it and write down any questions that you have. Talk to you later.